Hi, good morning. Um, I want to believe you are all doing well and settling into the class by now. This is chapter 1, and I will try to do some short videos like this on some chapters. If you also feel that there are some aspect of a chapter that you want me to teach particularly, it's okay, you send it. Sometimes I avoid sending a bulk of like 2 hours, 1 hour recording to avoid. I know most of you may not, it may not interest you if it becomes too long. So I'm going to do it short. Now, I will focus more on the aspect that I feel that needs to be recorded. But it's you are required to look up everything, but I'm focusing more on the part that I know I need to explain. To reduce the longevity, the length of the video, I will only focus on the part that needs to be explained. The part that is self-explanatory, I'm going to just leave it. So chapter 1 is the study of life. And again, the textbook you are required to have is the Open STAX, and I already posted the link for you on Blackboard. Now, we'll be looking at what is biology, and I will also focus on the process of science. Characteristics of life, we can look at it briefly, organization of life as well. All right, now, what is biology? From the word bio and logy, anytime you say logy, it means life. And from the word bio, anytime you see logy, it means study. So when you see zoology, anthropology, pathology, physiology, anytime you say logy, it's a Greek word that means study. And bio means life. So that means biology is the study of life. It's as simple as that. Now, if you want to make it tech, you can say it's a scientific study of life. But you know that it's all about study of life. All right. Now, why do you study biology? Obviously, you study biology. It's a base, since it's the study of life, then it could be a base, it could be the bedrock or the foundation for all inventions that are made, either in sciences, through pure sciences, engineering, medical sciences. Biology still stands as a basic foundation for, you see, biochemical engineers, biomedical engineers, all of those engineering sections that are committed to inventing devices, all of that, they are all linked to biology. Chemical engineers, they need a basics of biology. Biomedical engineers, they need the foundation biology to be able to function well in whatsoever they do. So all of those all of those people that invent all of these devices, they need the concepts from biology to be able to do it. Biology also helps you to make informed decisions about your health, nutrition as well. Most of the new breeds of crops, produce that are made, they are all from the concept of biology. Biology also open up more career. I'm going to go away from this, but this is interesting, cyanobacteria. If I want to go about this, I'll be talking a lot, because I'll be talking about how the heart existed billions of years ago, and it was only prokaryotic organisms that are able to survive at that time because there was no oxygen and because of the level of radiation as well. And how cyanobacteria later came into place and they started oxygenation, which was what later gave eukaryotic organism privilege to live. So about 1 billion years when prokaryotic organism began to exist, there was no eukaryotic organism. because eukaryotic organism cannot survive the rough and unconducive environment we had. No oxygen, eukaryotic organism cannot survive with that. Radiation was too much, it's going to kill DNA, it's going to destroy DNA. So eukaryotic organism cannot survive in that atmosphere as well. But cyanobacteria was very helpful in the in priming the existence of eukaryotic organisms. So I won't, if I get to that, I will be talking a lot. So let's go. Now, process of science. Science, meaning knowledge, covers general truth when tested and by scientific method. Now, when you say you are talking science, you are talking specific truth. You are talking about proven truth. You don't talk about science and you are saying, I think. No, you talk about science with reference to work that has been done. Now, when you are saying that this is water, and you are saying the properties of water, you must be able to tell us why is that. When you say water have adhesive property, like this now, you should be able to explain why does it have adhesive property. It's not I think. There's no thinking in science. Science are facts. They are truth. Proven ones. So let's go. How do we come across science? How do we come across proven truth that you can relate with, that is the same today? If it's the same in United States of America, it's the same in England, it's the same in Canada, it's the same in Nigeria. It's not going to be different anywhere because those stages are definite, they are specific, they are proven, they are testable, and they are repeatable. Now, if I have to perform an experiment here in the US, it's going to be the same protocol as it is going to be in Nigeria too, regardless of the weather. So how does that come into place? That's what we'll be looking at shortly. Now, I will go away from this because I'll be explaining it. Now, scientific method, when you say scientific method, you are talking about method of research that have definite steps which is going to lead you towards a result. The result can later be published when it is formulated. Now, scientific method is very important. No scientific method, no science. I could say that if there's no scientific method, there can be results that are proven, that are definite, that have steps that are reproducible. Now, I will just go straight to, let me talk about hypothesis. No, I will come back to hypothesis. This is what we call scientific method. When a scientist see a problem, a scientist observe the problem. And that's critical for every problem solver in the world, even if you are not a scientist. Everyone that will solve a problem must first observe that there's a problem. If you don't observe that there's a problem, you cannot solve the problem. It's only a problem that you observe that you can provide solution to. Now, a scientist are very observant. Now, when you notice there's a problem, then the next thing is to ask a question. Now, this is my car keys remote, and sometimes ago the remote was not going to work, or sometimes it works, sometimes it doesn't work. Then I ask, what is the problem? So the first thing is I observed that it's not working efficiently. Then I ask, what could be the problem? The first thing that came to my mind after I asked the question, what could be the problem? So the question is, what could be the problem? The first thing that came to my mind, which is, can it be the battery? So I can form an hypothesis from there, which is what I'm talking about now. Hypothesis is, it is the battery. Or if I change the battery, then the remote will start working. That is an hypothesis. If I change the battery, the remote will start working. Now, that is an hypothesis. It is now my responsibility to prove if the hypothesis is true or it is false. I am saying if I change the battery, the remote will start working. Then I went to AutoZone. I told them I need a battery for this remote. I gave them the remote. I opened it, got the battery, I inserted the battery. I have performed an experiment. The moment I finished installing the battery, I went back to the car, checked all the features, everything works perfectly and more efficient. That means I form an hypothesis based on what I observed. I performed an experiment which was going to AutoZone and changing the battery. Then my results, the remote works efficiently now. That means my hypothesis is correct. Then I can go ahead and report the results and say, my hypothesis was correct. Remote wasn't working. Now I can formulate a theory that when some buttons in your remote are not working efficiently, or when your remote works on and off, then go to AutoZone and change the battery. That is my report. Now, if on the other hand, I change the battery and it's not working, it means that my hypothesis is not correct. Then what do? I can go again and form another hypothesis. Now, if my remote still didn't work after I changed the battery, then I'm going to form another hypothesis. Now, my hypothesis can now be change the remote, go to Toyota and buy another remote. Then I will test that again. Then if it is correct, it means that the remote was what was bad. A new remote works. That is simply what experimental scientific method is. It's what gives you experimental design. All right. Now, this is still another analogy which you can. Now, hypothesis is the most important thing here because no hypothesis, no experiment. Now, what are the things that are very, very important about hypothesis? Now, hypothesis is not just a guess. If I take this remote to, I'm from Nigeria and I'm from western part of Nigeria, then I am from a remote area from that western part. If I take this remote to an old man in that remote area, the man will not know what the problem is. He will not even know if there is any problem with the remote. He may just be excited that he has something to play with. Now, the man may treasure it, but the man doesn't know what it's used for. If the battery is not good, he doesn't know. If the remote is not working, he doesn't know because he doesn't know. Now, that tells you that hypothesis is not a function, is not founded on ignorance. Hypothesis is not founded on I know nothing. Hypothesis is not just a guess. No. As some people have said is an educated guess. That's close to it. Right? Hypothesis is an informed thought on something. You are studied about something, you know it too well, and so that's why we come up with the issue of educated guess, meaning that it is a proposition, it is a thought that is coming from being informed. If you are not informed, if you have not studied about something, if you don't know something, you can't make a right hypothesis. So hypothesis is not just a random guess that, maybe this is something that is wrong. No, it's something you can say that for the time I've used remote over time, if something is wrong with the remote, then it will most likely, the first thing I want to check is going to be the battery. then some other things can come into place. Now, so that is what an hypothesis is. So hypothesis, correct hypothesis, is going to be based on having a background knowledge on the topic you are talking about. So if you are talking about coronavirus, for instance, and you are proposing an experiment, you want to design an experiment, you are going to come up with an hypothesis first. Now, but before you come up with an hypothesis, you will have studied previous works about it. So your previous knowledge is what is going to inform the hypothesis you want to make. Because hypothesis should be true, but it may not be true. Now, it should be true because it's something you can explain with science, what you are saying. Your hypothesis must be visible. It must be logical, because you can put science together and say, if antioxidant is able to do this, then it can reduce DNA damage. You want to look at that, then you are saying if autophagy clean up the body by itself, then it can remove some dead cells and it may prevent some things. So by the time you are saying those informed knowledge, that is, by the time you are looking at your previous knowledge of something, then you can make an informed decision or informed thoughts, which we call hypothesis. Now, every hypothesis that you are saying must be something that you can test. You can't make an hypothesis and just conclude a theory from there. No. Hypothesis must be something that will be experimented, so it must be testable. You should be able to perform an experiment on the hypothesis you are projecting. Now, it must also be able to be proven wrong. You can perform the experiment and say, okay, based on this experiment, the hypothesis was wrong. Then you generate another hypothesis. And that's why a lab work can take a longer time. Because think about it, you have an hypothesis, you run the hypothesis for about a year, then you realize the hypothesis was wrong. Then you have to form another hypothesis and continue again. So then you have variables. Now, variables are the factors that are unknown. Now, if you say that Tylenol is going to, will Tylenol be the best one? Now, if you are looking at, yes, I can use, let's use Tylenol. Now, if you work all day, all night, and you have to come to my 8 a.m. class, if I have 10 students that do that, then I can say that everybody knows that sugar increases people's activity. Then I can say, okay, I want to see the people that will be most active at that time among the 10 students that have gone to work. I know that sugar increases normal activity. So let me give one of them orange juice, chocolate, package all sugar for one of them. Then let me have 8 of them that I give different concentrations of Tylenol. Then let me have one of them that does not take anything. I just give the person water. So one is water, one is sugar, any sort of sugar, chocolate, orange juice, mango juice, all of that. Then I have 8 which I call treated groups. So you call those 8 treated groups. They will have different concentrations of Tylenol. That will be my variables. So the 8 that have different measures of Tylenol, different concentration of Tylenol, will be my variables. The 2 groups that have orange juice, sugar, and also water will be my controls. Now, I have 2 controls. You should take note. Now, one of the control is a positive control. Positive means that I know that sugar raises everybody's activity. It's default. There may be exceptions, but normally sugar will always raise everybody's activity. Some people, it is coffee, right? Now, that is my positive control, because I already know that sugar will raise the concentration, the activity level. That's my positive control. So what I'm expecting to see in that guy, even though he had to work overnight, if by the time he comes to class, I give them sugar. And I've done that for one of my classes before. I had a 6 p.m. class, then I take chocolate to the class every day, because I know it will raise their activity level. Now, but I also have another group that I only gave water. So the group that have just water, I don't expect water to raise any activity. I expect that to just make them the way they are, tired, right? So that is my negative control. I don't expect any result from them. I expect that they will be normal, like tired. If you had gone to work overnight, you should be tired. And they will all the 10 students will have the same traits. They will be about the same age. They will be in the same class. So they have common features. I can make this like 2 students. And normally I can make this like 2 students as well, so that it's not just one, right? Because I will be able to look at both of them. Both positive control are controls that you have known over time what their result will be. If you do this, you will get this. Those are positive control. Negative control is you know that what you are giving them does not have any effect on them. As a matter of fact, the reason we talk about water is just to give them something to survive, not because it will have any effect on them. So that is a negative control. So anytime you are administering something to a group and you know that it will not give them, it will increase the activity, it won't change anything about them, it won't prove what you are trying to say, then that is a negative control. But anytime you are performing an experiment, but there is already another medication that will also do what you are trying to test, right? It is a positive control. It could have been another scientist that have performed the experiment, but it's already proven. Now, everybody already know that sugar will raise people's activity, or sugar will raise high concentration of carbohydrates, all of that will raise your blood sugar. So if you are trying to look at an experiment and you want to see if eating a particular food will raise people's sugar, like let's say you want to check if eating protein will raise people's sugar, but you already know that carbohydrate will normally raise people's sugar. So you give some control group carbohydrate, then you give your normal experimental group. Now, experimental groups or tested group, they are the group that only have what you are trying to test. Positive group, positive control, you will administer what has been proven before by other scientists over time. You know it will give a positive result. It will give a change. But negative control will not change anything. It's just, now the reason you have negative control is so that you can compare this group with your tested group, or with your experimental group, or with the variable. This will change, but this will not change. Nothing will happen here. So if nothing happens here, by the time I finish my experiment and the students that add water is not supposed to be active, so good. That means they all went to work and they should not be active. Then these other 8 guys that went to work and I gave them different measures of Tylenol, if all of them became active, then I will know that my Tylenol worked. Or if some of them became active, then I will know that, okay, maybe it's this concentration of Tylenol that works for this age or this class of students. Now, but I expect them, the positive control, to be hyperactive, right? Good. So that's the difference between positive control, negative control, and variable. If you have questions about any of them, please feel free to ask me. I'm trying to explain as little as very clear and simple as I should. But obviously, I don't have a feedback. So if you have questions, please shoot me an email. So I've explained this. I've explained negative and positive control. This is just an analogy for scientific investigation. Please follow that. We've gone through this. I've talked about positive and negative control, right? Now again, I'm going to run through it. Positive control it's just, let's say we are talking about medication, but there was a medication you are trying to, so let's say my name is Alonge, right? Then I just come up with a medication that is called Alonge hyperactive. Nothing like that, right? It's not going to come. Alonge hyper. Then I'm saying it will make you active. It will increase your intellectual activity. It's Alonge hyper. Anytime you go to class, you become hyper. You become active, even if you were tired before. That's the name of my medication that I want to do. But now that medication is unproven. Then I need to get a medication that is proven so that I can be using it to compare the effect of my medication on my subject. So if I put 10 people in class, 5 people now, if I give all of them my home new medication, how do I tell if my medication is having an impact or not? No. So I'm going to have a control, which is the positive control. I already know that if I give them Tylenol, it's going to refresh them. Or some people, I know if they take coffee, it's going to refresh them. So I can make a control that just give the student coffee, students that are proven to respond to coffee very well, I can give them coffee and make that my positive control. I can have another positive control too and give them Tylenol because I know it will refresh them. That's another positive control. So what is common about positive control now? I know that there is an effect when there should be an effect because Tylenol is going to increase the activity. Coffee keeps people awake too. So now, if by any chance the positive control does not produce the expected result, I know that maybe there is something wrong because I know the positive control is proven. But there's no way I can tell that my result is proven because it's not proven yet. So if I have an experiment and the positive control and my result does not produce anything, then I know it's not about my result. It's because positive control that is already proven, is already tested, is not performing the results. So I will tell maybe there was something. Maybe there was a general season that had effect on them. Maybe it's summer and everybody is hot and that's why their performance is low. Something like that. Anything could have happened. But on the other hand, negative control, it's sure that nothing will happen. That's why we use like neutral substances, maybe like water. You know, water will have no effect on people except that they had to pee, right? All right. So negative control does not have, you don't expect that it's going to have any change. All right. So this is more explanation about negative control. Watch this video, it will give you more explanation about positive and negative control. Then when you are performing an experiment, you have to have a good design. Now, a bad design is going to be to have an experiment that you don't have control groups. That's a bad design because how do you convince us that what you give, the medication you are administering is performing anything? There's nothing. Now, when you are doing DNA study, we talk about markers. When the first thing you put in your gel is the marker. The marker is going to be the control because the marker is already proven. Proteins or DNA, they have different weights. So the marker is already proven. So if you are trying to check for a particular DNA or for a particular protein, anyone that comes close to your marker will be what you are looking for. Since your marker for the DNA you are looking for, let's say you are looking for you are just trying to check some antioxidants and you want to check catalase. Now there will be a protein, catalase will already have a marker. So you are going to get a marker from a standard lab. Then you are going to put the marker as the first thing on your gel. Once you put the marker stays on the size where it's supposed to be. So let's say the protein is like 50 kilodalton. It's going to stay, the marker is going to stay there. So once the marker is there, you know the marker is going to stay right on 50 as well. So let's say this is 50. The marker is going to be around here. So you notice that you have this here. So this is 50 kilodalton. Now this is going to be here. So if this is your first well, which depicts your marker, then you have other wells. Now, what is going to happen if you put some of your substance on the first well, and everything goes here, you know that's not the protein you're looking for. You put another one here, everything goes here, you know that's not what you're looking for. Then you put the third one, and it's here. Now, then it tells you that this is the protein you are looking for. Then you may put the fourth one, and it's here, but very tiny. So that tells you the protein you are looking for is here, but it's just a tiny bit of it, right? Now, if at the time you, so again, this is your control because it's the marker. That's why I turn it to red now. So that's your marker. So is the control. All of these 4, 1, 2, 3, 4, they are experimental group. So an experiment that doesn't have a marker is not a good experimental design. I mean, that doesn't have, yes, a marker when you're talking of DNA or protein analysis, electrophoresis. But for general experiments, if an experiment doesn't have a control, either a positive or negative control, that is not a good design. Your variables too should be, now if you are performing an experiment, now look at this. For this experiment, I have 4 tested groups. I have 1 control and 4 tested groups. Now, this is good because I have 4 tested groups. So I can have this, and this to look at. But if you do a tested group and it's 1 can happen by mistake, right? Because even after I've done this experiment and it's all gave me the same result, I should do it again. Usually, you do an experiment 3 times. That's why when you see a bar chart, sometimes you see an asterisk on it or a star. It means that the result you are seeing there, sorry, I'm not able to do a star very well with this. It means the result you are seeing is an average of results. It means even though they have just 1, but it's an average of some results, meaning that the experiment has been done more than once. That's what it means. So you must have the test group, which is, this is my tested group now, and you must have the control. The model must carefully represent the object. Now, so sometimes when you want to do some experiments, you use, now if I want, you use human, you use rats, you use like rats, you use rabbit, for some experiments to test some medication that they will later use on humans. The reason they do that is because human and rabbits, they have both, they have similar compartments that if it works on them, it can work on human, right? That's why. Now, if you want to test some medication, you don't use some medications on bacteria and think it will work on animals, on eukaryotic organisms. No, because bacteria is not, they are not eukaryotic organisms as human. But if you test it on some viruses, on some virus, then it can work on animals. And that's why viral infections are difficult to treat in human, because what the medication you give to kill the virus will most likely affect human because they are both eukaryotic organisms. But bacteria is easier to treat. Then after you've proven you've checked your experiment, it's positive, then we go ahead to present our results. So let's go. So I've talked about this again. I've talked about this. All right. So sharing the information is about the results, which I've talked about. Is either you get it to a paper, you publish a peer-reviewed manuscript, or a scientific paper, so you can just read that. Now, what does it mean to be alive? Properties of life. This is like a short way to learning. Could Susan's mother have golden hands? Now, if you take note, the fourth word comes in place here, and that's what you can always do. You can look at first words of every word to make something that you can always remember. Sometimes when you remember the first word, you can remember everything. So the lowest or the simplest level of organism is cellular. The characteristics of organism. We are going to get organization of life. Don't mind me. Once I saw cellular organization, I thought I'm there. These are characteristics of living organisms. If a cell, the simplest level of life is cell. If you cannot trace a cell in an organism, a claimed organism, then it is not a living organism. Cell must exist before a substance will be called to have life. I hope that makes sense. Then obviously you should be able to respond to stimulus. If I pinch you now, you should respond. That's life. If I pinch a dead body, it does not respond. That is not life. Metabolism, it is, you should be able to respond to metabolism, catabolism and anabolism, build up and break down. Homeostasis, you should be able to maintain internal, regular or an internal environment that is conducive for life, right? Then growth, obviously, and reproduction, very important. Also heredity. Please watch the video here to give a clear grasp. All right, so I will go through the level of organization and I'll be closing there. All right. Now, the basic level of organization, the simplest in the level of organization is organelles. Sometimes you see chemical level of organization. That's still fine. Chemicals form organelles, right? Now, when you say organelles, don't confuse organelles with organ. Organelles, they are smaller organs. Think about it that way. They are not organs. Organs are larger. Organelles are small. Now, they are smaller. So you can see organs here, right? These are organs, but these are organelles. Organelles are tiny structures within a cell. When you see a cell, like plant cell, animal cell, for example, you have organelles. A cell have the cytoplasm, which is the fluid area, and also inside the cell, at the center of the cell, you have the nucleus. Now, the nucleus is what we call what hosts the DNA, right? Outside the nucleus is the cytoplasm, which hosts organelles. You have the ribosome there that makes protein. You have the Golgi apparatus there that help in processing and packaging of substances. You have the endoplasmic reticulum, the smooth and the rough. The smooth makes lipid, this rough makes protein. And why does it make protein? It has some ribosome on it, right? You have some cytoskeleton. All of that, they are organelles, right? You have mitochondria, they are organelles, right? Now, they are sub-organs inside a cell. They are smaller organs inside a cell. So I can call organelle cell's organ, right? They make up a cell. Then cell, you know what cell is already. You have the human blood cell, white blood cell, all of that. They are made of from organelles, right? Then tissues, cells that work together to perform a specific function. So tissues are bigger than cell. Then you have organs. Organs, they are larger than tissues because they are tissues that work together to perform a specific function. Then you have organ system. Organ system is when you have different organs that comes together to perform a specific function, right? You have the the respiratory system, you have the digestive system, you have the urinary system. So there are different organs There are different organs in those systems. That is, you have respiratory system, right? So there are different organs in those systems. Then organ system makes organism. Don't forget, organs. Your heart is an organ, but it's inside the respiratory system. Your lungs is an organ, is inside the your heart is an organ, is inside the cardiovascular system. Did I say? Then inside cardiovascular. Now, from the word cardiovascular, that was a mistake. Organ, what I said about heart before. Now, heart is inside cardiovascular system. Now, cardio is heart, vascular is blood vessels. So that means in cardiovascular system, you have cardia, which is heart, you have vessels, which is vascular. So you have blood vessels like artery, veins, blood capillaries in the cardiovascular system. So you have different organs inside the organ system. All right. Now, organ system, different organ system, respiratory system, cardiovascular system, urinary system, digestive system, the whole lymphatic system, they all work together, and that's what makes an organism. Then different organisms work together, it forms a population, different population, community. And obviously everything in life, microorganism, all of those plants, all of that, they all form ecosystem. And everything, the heart, everything in the heart, it forms the biosphere. All right, so that is that. Now, theory of evolution, which is the last thing I would look at before I go. Now, this is my acronym for evolution. I'll just give you the acronym. It's BAE. Now, when I say this, I just tell my students, domain is bay. Domain is beautiful. Domain is bay. Now, B is bacteria, A is archaea, and E is eukaryote. So domain is bay. Now, we have three domains in life. We have the bacteria, archaea, and the eukaryote. These two, they are prokaryotic. I'm not sure I should go to this now, but it's fine. These two are prokaryotic organisms. You can call it prokaryotic or prokaryotes. And this, obviously from the name, is eukaryote. When you see pro, anytime you see pro, it means they are before. So I'm coming. Now, this karyote is from the word karyon, and it is used to depict the nucleus. So prokaryotic means they are before the nucleus came into existence. So they don't have a true nucleus. But eukaryotics, they have the true nucleus. And this is where you belong. You are in the animal kingdom, right? Now, but these are the domains, bacteria, archaea, and eukaryote. Now, okay, so those are the domains. They are those bacteria, archaea, and eukaryote. Then those are you have the kingdoms there, all of that. Let's go. Now, this that you have here, we call it phylogenetic tree. Phylogenetic tree means it is. So we can call it phylogenetic tree or evolutionary tree. Now, it's just showing you the tree. Evolutionary tree or phylogenetic tree shows you the evolution, organization of animals, of plants, all of that. It tells you where they belong, which of them belong to the same family, which of them belong to the same, have the same descendants, and that's clearly what you can see here. So that's just what it shows you. That's what phylogenetic tree or evolutionary tree tells you. It just shows you their ancestor, if it has common ancestry or not. All right. So I think I'm going to stop here now, but feel free to send me questions if you have. It's becoming too long already. So have a very good time. Listen to this, ask questions. But those are basic things that I need you to know from this chapter.